you're in one of these stands, you're looking up, you can see the defoliation, sort of, especially at the tips of the branches. This is probably an example of multiple years of defoliation, but it tends to feed on the newest foliage, but after multiple years, it gives this sort of skeletal look to these trees. And ultimately, you know, 6-7 years of severe defoliation like these have sustained is when your trees start dying. And if you look at over sort of a broad spatial scale in terms of outbreaks, this is defoliation from the 30s up to 2012. You can see there's budworm outbreaks of some kind throughout Canada at any given time, but especially you can see in the East Coast, we're about to come into the major outbreak of the 70s and 80s, and that one was almost 55 million hectares, so a monstrous size outbreak. And the question that comes up when we look at these different outbreaks, one of the first questions I often get is like, are these budworm outbreaks just a natural part of the ecosystem, or is this something that's caused by sort of human intervention? And we do have evidence to sort of look back pretty deep into time, almost back to the last ice age, especially in the paleofossil record. So we can go into these spruce peat forests. This is a picture of a shoot. You can see the little lumps of that's frass or budworm poop on there. Those drop onto the ground, and those are basically preserved in the layers of peat. So years and years and years, we can go back. And if we take a core from these, as some of my colleagues did up in Quebec around 2006, you can get this really nice sort of time capsule of budworm outbreaks reaching back, in this case, almost to 8,600 years into the past. So it's sort of a present at the top all the way down to the oldest at the bottom. And you can see these are basically measures, these bars here are measures of, use my arrow, these are measures of densities of budworm frass they've found at different times. You can see one around 6,600 years ago, so a little bit of something of a spike around 8,000 years ago. And there's the present, just to put these time numbers into perspective. Oh, sorry, there's the evidence for budworm, or the time for budworm. You know, this sort of includes just basically not long after the last woolly mammoths went extinct, but before the pyramids started being built. But really all the information we have for budworm, all the detailed information comes from just this top little period of time of the last few hundred years. And we can get this from other sources. So that gives us really crude data telling us that budworm has been around for a very long time, probably defoliator in trees, long before we were interested in forestry. This is a much more fine scale look at what those outbreaks actually look like. And we can get these from various sources of data. We can use tree ring data. These sort of shrunken periods of tree growth in old timbers can give us indications of when spruce budworm outbreaks occurred. More recently, where the lines start becoming solid here in this time series, we usually get this from things like defoliation maps and on-the-ground collections, where it gets really jagged. Those are on-the-ground collections where you get very detailed data. But you can tell it basically tells you one continuous story. And we've been doing these for the last 50 years. You can see, I mean, the only thing that's really changed, we have more technology for using GIS, and the fashion sense has changed maybe a bit, but in general, we're basically collecting the same data. We've been doing so for the last 50 to 100 years. And the striking thing is that we have this really strong periodicity of these budworm outbreaks, you know, every 35 to 40 years in the cycle. And of course, this begs the question of what exactly is causing these cycles to occur. Now, there's multiple hypotheses that have been sort of put forward to explain this. I'm just going to talk about two of the main ones that have sort of come up in the last 50 years, because they tend to have been pitted against each other. During the 1980s, this sort of driving hypothesis with this, we call it the oscillatory hypothesis, was this idea that there are predator-prey cycles that essentially drive budworm outbreaks. And this is very similar to other ideas at the time that were looking at these lynx hair type of dynamics. So what this looked like, basically you have this sort of characteristic of your first year biology. You have the blue lines, which is the hare populations, they're coming up. And of course, the predator populations trail that. And as the populations of the hare peak, or as the lynx peak, they pull down the rabbit population. And when the rabbit population gets low, so too does the lynx population. And that's, you basically have these sort of dual cycle of the predator and prey going forward. And that was really how they interpreted the spruce budworm. Now, it was a little bit different because the predator involved in this was a variety of different parasitoids. In this case, this is a pupal parasitoid, but there's all sorts of ones that also attack the larvae as well. And so I looked at this basically as a dual predator-prey cycle that was moving these budworm outbreaks forward. The problem was that this was based on an interpretation was just from data, like detailed population data we collected from the tail end of outbreaks. You know, we never really got good, or never really had good data from when populations were rising. So, another sort of interpretation of this hypothesis as it came forward, this is actually for the outbreak from the 1968. I've skipped ahead years as we go here, but it sort of started around the 68. There had been budworm through New Brunswick throughout this time, but at relatively low densities. This is 1968, jump forward 3 years. Those 3 patches had started to increase. By 1973, you're starting to see them coalesce together. 1975, which was the peak of the previous outbreak, it was the highest year, I think, of defoliation. And you can see the whole outbreak is basically coalesced together in one sort of continuous block. And the implication for this, we went from these 3 patches to a broad scale population. And the argument that was made in part from this was that moth dispersal really didn't contribute to spreading outbreaks. You had basically a regional population that was governed by these predator-prey dynamics, and all the populations were coming up and diminishing roughly at the same time. And you had moth dispersal that was maybe contributing to increasing the intensity throughout the region, but it wasn't really causing populations to either rise or fall. And in this sense, if you have this sort of large-scale population increase in this way, these outbreaks were viewed as being inevitable. And this had implications for how we manage spruce budworm. Now, moving forward to the current outbreak, now of course there has been all sorts of research that was done during the last 30 years. But if we look at just very roughly how this outbreak has developed, you can see it sort of started in a few patches. Those increase, coalesce. And there's a much clearer pattern. Now, we have much more detailed data, and there's a much clearer pattern that we would interpret as spread. And this was consistent with some of the ecology data, some of the ecology data that had been collected and some of the experiments that had been run. This started to give us this idea that, first of all, predation didn't seem to actually decline before the outbreak increased, which is, of course, one of the requirements. This idea of a predator-prey cycle involves the predators declining to a point where budworm is released. We weren't seeing that in the actual data we were collecting as populations were rising. It was almost unsustained predation. And the other thing, that there was a much stronger influence of moth dispersal that was actually helping to spread these outbreaks. And our conviction with this has just been increasing over the years as we've sort of been watching this outbreak develop. So this led to sort of pushing us back to the 1950s. I saw Gordon Mott was online. This is part of the work he was doing. It was called the double equilibrium hypothesis, and it offered an alternative explanation for how these populations rose and spread. And it was much more, best described maybe as outbreak spreading contagiously. You know, basically hotspots rising and seeding new hotspots down the horizon, sort of like chickenpox might spread through a classroom. There was at least some element of the budworm outbreak that involved a spread component to it. And it doesn't mean that it just completely offset the oscillatory hypothesis. There are still many parts of that that were held firm, but at least as the populations are rising, it seems to be like some added dynamics to that. So here's roughly where we are. This is 2015, so I'm a little behind on my maps, but so if we have these two hypotheses, the oscillatory hypothesis gave rise to this idea that it's an inevitable outbreak, so we should basically just be treating small patches, high value stands within this area. There's nothing we can do to stop the outbreak or to slow the spread of the outbreak because it's just rising everywhere regardless. But with this new interpretation where there was something of a spreading component that was perhaps at least partially driven by moth dispersal, led to the sort of rise of this so-called early intervention strategy. And this is really just a containment strategy where we're trying to work along the northern borders of New Brunswick, treating hotspots to hopefully slow that spread south until the outbreak collapses naturally. You're probably familiar with this description. It is very similar, at least conceptually, to the slow the spread program for GYPSY moth. GYPSY moth is, of course, the slow the spread program is where they're trying to slow the westward spread of that. It's an enormous program, frankly, but it seems to have been fairly successful in terms of slowing the spread of that outbreak. There are, of course, differences ecologically and biologically. It's a very different insect. It's also an invasive, so it has different sort of dynamics within the environment. But at least conceptually, this idea of containing and slowing the spread of an outbreak is something that we've tried to capture in the early intervention strategy. So So what I'm providing here, these are basically fairly consistent for any kind of containment strategy, but these are the criteria for feasibility for actually making a program like this work. These are the pieces that you need to implement a program like this. So the pieces that we focus on in our research trying to develop to make this program work. Of course, the spreading dynamic is a big part of that, and I've sort of covered the population dynamics aspect of that. The other part, really important part, it's the starting point for any management program, is of course the monitoring and the ability to detect populations of low density. And Allison did a really good job of describing how Maine is developing their own, their work to some extent in coordination with us to develop this ability to detect at low densities. And she's obviously offering evidence that they're finding them. So just as a reminder, this is as of the spring 2017. This is the defoliation map as of that year. You can see that the defoliation is creeping right up on the border of New Brunswick. There's a little bit peppered across, sort of like maybe Allison's seen. There's a little bit of defoliation we're seeing across the border, but not a ton at this point. Now, defoliation is one way you can look at this, but it's not great for detecting really low densities. And the best way for doing that is by detecting the L2. Now, that's the overwintering stage of the larvae. Here's a picture of one of the tiny L2 larvae. It's just come out of an egg. They're very tiny. They find the cracks and crevices in the trees, and that's the stage that they basically spend from August all the way through to the following May or June, depending on where they are, and hibernation. So we basically take those branches, we collect branches all throughout that period in Maine as well, and we wash those and we use that as to estimate densities. And here's one of my colleagues doing some bullet printing here to collect them. I'm, as Allison said, I'm sure many of you have done this before too. And at least in New Brunswick, so this is the kind of map you can get from that. So you can see all of the dots on there represent a point where they've collected 3 branches, and those branches have been washed to get the L2 off and to provide a density per branch. And so I'm just going to remove the points that were relatively low density, and that leaves us with just, you can see these clusters of stars along the border, the odd points here and there. These are all, by the way, over 9 per branch, just to give you a little bit of perspective. So anything less than that, we're not interested. You start getting in red, you're looking at 20 to 30 per branch. And as Allison said, we've gone through 75% of the branches in Maine, and we found 3 total larvae. So you guys are still very, very early compared to where we are. But what we do is we take this patch, or this sort of network of points we have, and we use some interpolation, some GIS interpolation, the royal we, my colleagues do this, I don't know what I'm doing for that. And they do this basically interpolation, and that's what they use to basically set the spray maps for the next year. Okay, and these, so basically within these, they set the lines for where the planes are going to fly. So the purple is represented tebufenicide. That's one of the insecticides that we have registered. The yellow represents BTK, which I think some of you are probably more familiar with. And so you can see we've got a bit of a patchwork that, if we look at 2017, it basically overlays these areas where we found these densities. So this is how we sort of set where we're going to treat for these hotspots. And it seems to be a fairly effective approach for monitoring. Now the question is, how effective is that for actually managing the budworm population? How efficacious or how effective is this? And are we having non-target effects? So this is where my expertise actually does come in. I'm a population ecologist. I'm interested in understanding factors that influence why populations rise and fall in outbreaking insects. Each of these little dots here represents one of our sites where we're collecting anywhere between 15 and 30 branches. You can see we have a network that overlays the area where treatments were taking place, at least in eastern New Brunswick. And we have a lot of transect of those that runs all the way down into Cape Breton Highlands. Excuse me. Total 70 sites. We're doing detailed sampling multiple times per year. We get our sort of L2s at the beginning and the end of the season for annual survival. And we get our seasonal survival. We're collecting periodically throughout the year before treatments and after treatments of insecticides. And fortunately or unfortunately, it wasn't planned, but we ended up, because there was a spate of really bad weather right in the middle of our treatment period, we got it to also test whether doing an early treatment or a late treatment with the insecticides almost all the way to L6 is effective for suppressing these populations. By the way, the data I'm going to be showing is really just for this area up in the north here, because we found no budworm essentially all the way down in the south. So the answer for those ones is 0. So if we just look at mortality, so young larvae to adult, that basically encapsulates the period of for initial feeding, treatments of insecticides if they are used, and all the way up to adult. And we're looking at percent mortality. And if we look at untreated sites, percent mortality, at least within this period, was around 65%. Now, if we look at the treated sites with all of the sites that were in treated areas, BTK or mimic, I've just pooled them together here, you can see the mortality was almost approaching 90%. But if we pare it down to just the ones that were treated early, we're getting slightly more mortality. So this at least gives us a hint that treating, I mean, some of the sites we were treating late were almost ready to pupate. So I'm fairly certain that we had some sites that just really didn't have much mortality associated with the insecticide. It was just, unfortunately, the timing for those ones was just a little bit too late. But still, even in that case, we had a pretty reasonable efficacy. And for the biologists out there, this is really a good measure of additive mortality. So this is mortality that's over and above what would be occurring during the season. And this is one of the things that we aim for in this type of program, is trying to get this additive mortality that wouldn't be occurring otherwise. Now, if we look at L2 to L2, so that's the way right at the beginning of the season, all the way to the end, and we look at this might be a little confusing, but it's just population growth rates. So if it's 0, that means the population from 2017 to 18 didn't change at all. So that's that dotted line there. If it's red, as it is in the untreated site, those populations increase. So we had generally increasing population growth rates in the sort of sites that didn't have any treatment. If we look next to it in the blue, the ones that were treated like both early and late, in general, most of them had negative population growth rates. So these populations declined from 2017 to 18. And if we treated them early, we had really good, every single one of the sites that was treated early had negative population growth rates. So that's very encouraging. And also, it's worth noting for this, this also includes when you have migration going into the area, because this would include whether you have both the survival effects, the effects of the insecticide, and migration going into the area. So that's not always the case. But we're going to talk a little bit about migration. And for that, I'm going to have to take you back to the summer of 2016, July 20th to 22nd. Donald Trump has just accepted the nomination from at the Republican convention for President of the United States. Canadians are watching their computers eagerly. Or we weren't watching the convention. We were actually watching the Weather Channel on the internet. Because what we've realized is that if you set the weather radar to snow, you can actually watch budworm populations, the moths as they disperse from the north to the south. So if you look at the little blue patches here, those are not weather because we have it set to snow, neige. So we have it set to snow, and we can actually watch the moths as they move south into New Brunswick. And so you can see this huge plume of moths as it's shifting south, and especially right over this Campbellton area. That's one of the northernmost towns in New Brunswick. You can see as it moves on, eventually those populations dissipate. And if you're downwind that evening, these are some of the sights you would see. This is an Irving station in Campbellton. You can see the masses of moths flying around these lights. The next day, you can go to the parking lot and you can see these mats of moths everywhere. They're covered in eggs. You can't see them probably in this picture for you guys, but there's eggs all over the place that they're laying. If you look at these bushes in the back, a nice close-up, covered in moths, laying tons of eggs on them. And these had a fairly large effect. So it was basically filling, especially in this area where we saw the largest impact. And it just so happened that we had sites that year that we were monitoring as time went on. So here's some of the sites. These were sites that were treated by BTK, these 3 red ones. We're going to focus on those and sort of look what happens from year to year depending on what's going on. So these again are just our annual estimates, L2 per branch. Excuse me. 2015, the site was treated with BTK. It was a fairly high density. It was pushed down to relatively low levels. There was basically nothing left. There's no defoliation and no pupae during the following summer in that site because we had been monitoring it very carefully. But there was that mass dispersal event, and those populations the next year spiked pretty substantially, up to almost 4 per branch or more. And this year, it was treated again with BTK. Those populations declined. And probably some of this contribution here is from migrants coming in. But the point is that this is really sort of encapsulates at a very small scale, this whack-a-mole sort of approach we're doing to this slow the spread up in the north there. There may be years where you have growth. We're going to try to treat those with the BTK or the tebufanicide, and we're going to see those populations decline. So far the results are pretty encouraging. And just to give you a before and after, so this is the L2 densities from 2017 that I showed you already. If we move forward to this past year, you can see that there's a little bit of spreading. It's hard to tell, 17, 18, 17, 18. The populations are pretty much the same as they were last time. Now, there was a little bit of spreading here down in this area, and these just happen to be the areas that we treated a little bit late. So we think that's probably the reasons. We just didn't really get good efficacy, but we got really good efficacy within this area. And that tended to push those populations down. And so probably a lot of, as we're sort of developing the mapping, a lot of our mapping for the trees this year are probably going to encompass these new areas that we have here. And so this is the game that we're playing in. But for the most part, we remained relatively stable from last year to this year, even as Quebec substantially increased in the lower St. Lawrence. So I think we're doing okay so far. But again, it was a low migration year, so we'll see how these things move forward. Now, that's efficacy. The other concern we have is, of course, with potential effects of using tebufanicide or BTK on non-target budworms. So this monster is the food web for spruce budworm. Where the yellow arrow is, that's spruce budworm. On either side of that, we have different other larval insects that are really closely related to budworm or distantly that are also feeding on the trees. And then we have this huge mass of different parasitoids and parasitoids that look for these parasitoids and hyperparasitoids, and just this really complex food web. This is what helps keep budworm really low during these long periods of time between outbreaks. And so we're very sort of conscious or sensitive to the idea that if we affect budworm as well as these other species, are we also potentially disrupting this food web and shooting ourselves in the foot to some extent for whether this is moving forward? We have lots of sample, all the stuff that we collect when I'm collecting branches during the summer. We also collect all the the other larvae that we find on branches, as well as some from other tree species we have in the area to try to get better look at this question. We basically freeze them, and some of my colleagues are doing some DNA analyses to identify the different parasitoids that we're finding, not only just in budworm, but also in these other species, and look at how these might change over time as we're treating these areas. These are my three colleagues, Alex Smith, Veronique Martel, and Michel Cousson, who are tackling this work. And hopefully we're going to see some more results from this by the coming summer. It's also worth mentioning that there's potential broader impacts that we think about the broader ecosystem. Budworm is part of the successional dynamics of forests. So it can help to thin out some of the older trees. It's food to things like specialists. This is a Tennessee warbler. It's a specialist of budworm. So you can go into forests that are having increasing budworm populations. And even before you really detect budworm, you can hear these guys chirping around out there. And sort of to flip it the other way, if you have significant budworm defoliation around some of your forests, around some of your watersheds, what you can end up doing is removing overstory that actually keeps a lot of these streams cool, and that can have negative impacts on the salmon. So it's not just a sort of a clear, it's sort of, there's a lot of different complex dynamics that are ongoing in a forest ecosystem when budworm's active. But I do think it's worth noting that with respect to a lot of the species of bird, which we sort of within our group, we talk quite a bit about. I mean, the point for a program like ours is not, and has never been, to eradicate budworm. It's really just to keep populations stable. And that's all we're really doing. You can see sort of as we push down in one area, they're coming up in another area. And our goal is really just to sort of keep these populations relatively stable up there. It's not really to sort of wipe them out, as some people get the impression for these. So there's definitely often insects in most of our forests, even as we are suppressing these populations, locally at least. So, excuse me. So we've talked about efficacy, something about non-target effects. What really comes up, though, is when you have all of these things can work: population dynamics monitoring, efficacy, excuse me, and those can fall apart if you don't get social license from the public and don't explain to them what you're actually doing and make them understand sort of the different aspects of these types of dealing with these problems, but of these types of projects. And that comes down to a lot of communication. And especially when it comes to around insecticides, that's always the biggest concern that people get. We get a lot of questions about them. To be fair, there's a lot of history around insecticides. We have, I'm sure, Maine's very own Rachel Carlson. She has a chapter in there, Rivers of Death, which was written about the DDT program in New Brunswick, of all places. I don't know if you recognize the young lady in the bottom right. That's the Canadian Jill Stein, Elizabeth May. She's our Green Party leader. And she cut her teeth initially working on, pushing back against the budworm Phoenicia work that was going on down in Cape Breton. So there's a lot of Canadian history that's built around pushing back against treating budworm outbreaks. And so we do a lot of work to try to, a lot of things have changed since the days when those books were written. And we try to do a lot of work trying to explain what it is we're actually doing and how we're trying to do this responsibly. The website is a good example. This is our equivalent to the Maine version, Healthy Forest Partnership. We're partners with Allison and Brian Roth and people in Maine. We share a border, and so we're partners in terms of trying to develop communication around what budworm is and what it's doing. As part of this, I write a research blog from time to time with some of my colleagues. We have our citizen science program that Allison mentioned, and I think there's some participants in this, which we very much appreciate. There is important scientific data that helps us better understand moth migration and how it contributes to movement. But part of this is also just trying to reach out to the public and talking to them about what budworm is, and like bringing them as sort of as community science into really asking questions about what it is we're trying to do with budworm, and seeing in real time sort of when budworm are coming into their areas. And I think it's very useful for drawing attention to the issue. We do a ton of media. That's me in the top right there, but my colleagues, bottom left is Veronique Martel, and Deepa's in the middle in the bottom picture. And, we do a lot of media. I think in the last 4 years, I've done about 70 or 80 sort of media type things. It's another way to reach out to the public, and this is, it's sort of an underappreciated part of forest management or pest management, that you do have to reach out to the public in all sorts of different avenues to do it. And I also give a ton of talks. Like this one. I think I'm over 130 or 40 right now for the last 4 years. One of the talks that seems to be especially well received is a talk about how the insecticides actually work. And I'm going to, some of you may know about BTK, but I'm going to give you a bit of a taste of what this type of talk looks like and how we try to explain what BTK is and how we use it. So BTK is essentially a soil bacteria. It's been commonly used for 100 years or so, though it was only more recently refined as a way of treating insects. And you see on the left these little sausage links. If you take one of those sausage links and look inside, there's really two components you need to understand. There's a protein crystal, and there's the spore, the reproductive organ, and the skin that surrounds all that keeps inside. That's all you really need to know about it. I actually drew this. This isn't a photograph for anybody that's wondering. This is a spruce budworm larvae eating some foliage, and those little guys there are BTK on the foliage, and these are gut bacteria. And so you're looking at basically its digestive system here. So when the insect eats, and this is any caterpillar, when it eats the BTK, those bacteria are taken into its stomach. The shell around the crystal and the spore are broken down. Those crystals embed themselves into receptors on the stomach lining, and they essentially lyse it, and they cut holes into it. And this is what releases all this, not just the BTK spores, but also the gut bacteria goes into the rest of the body. And this is what basically kills caterpillars when it happens. So the obvious question people ask is, okay, well, what happens if a human or any other animal I care about actually eats it besides caterpillars? To explain this, we have to dig a little bit deeper into how stomach pH works for animals. You don't have to understand the broader sort of biology of this, but most people understand that you have things that are neutral, that aren't very reactive with substances. You have things that are acidic, and you have things that are basic or alkaline that are reactive, but in completely different ways. So just as an example, we have water that's very neutral. It doesn't react with very much, obviously, nor would we want it to. If we're thinking about things that are acidic, bananas are mildly acidic. Vinegars are more acidic. Battery acid is very acidic. We're thinking about things that are alkaline or basic. Soap is mildly alkaline. Ammonia is more so. And Drano is extremely alkaline. You know, you don't really need to understand what this all means. You just need to understand that these substances or things that are acids are very, very different than things that are bases. So let's look at what's actually going on inside the stomach. It just happens that our stomachs of fish, deer, birds, humans, frogs, amphibians, most terrestrial organisms have extremely battery acid-like stomachs. And this is the environment in which our enzymes work. It helps break down our food. This is the environment in which we digest our food. If we move along that scale, we have things like shellfish. They're mildly acidic, probably because they live in salt water. For perspective, our skin is relatively neutral because we wouldn't want our skin reacting with everything. So that makes sense. And then we move to insects, and they have extremely alkaline or basic stomach contents. And this is the environment in which their stomach digests the plant diet, the largely plant diet that they have. It just so happens that for the skin to be dissolved around the spore and the crystal in BTK, it has to be in a very alkaline environment, sort of an ammonia level environment. And this is why it gets broken down in the stomachs of insects. Oops, oh, look at that. Oops. It starts to break down, even when it gets mild, like still alkaline, but moving towards neutral. BTK starts to break down and denature. It just basically doesn't function well in an environment. It comes from the soil, so it doesn't do well in even mildly alkaline or acidic. And so it starts to break down, and that's why it doesn't really affect these other things. You find it, and that's why it's so effective and so specific to insects. Now, there are things that are very happy to live in acidic environments, especially certain types of bacteria. E. coli, coliform bacteria. If you find a single colony unit of E. coli in your water, when they test it, they will shut down your drinking water. BTK, they won't even bother looking at. And so the analogy we try to use with this is sort of like, you know, every organism, as most people know intuitively, has its own environment, its own combination of conditions it requires to function. And this is the case also for BTK. It requires these particular conditions for it to actually be activated. This is what makes it relatively specific for certain things, but really sort of innocuous for others. And this is really the reason, you know, we say BTK is not toxic. This is the reason. The Environmental Protection Agency, U.S. Department of Agriculture, the World Health Organization, they've all said, some paraphrase of this, owing to the mode of action, which I've just described, BT products are unlikely to pose any hazard to humans or vertebrates, et cetera. This is the reason why it's used and is registered for organic farms. So if you eat organic produce, they had problems with caterpillar pests, they probably used some BTK for it. It's been used in downtown areas. It was used for forest tank caterpillar back in Winnipeg in 2015. And so it's fairly broadly accepted. Now, that being said, you know, we've had a lot of engagements with groups here, and it's one thing to explain this, but they really, if we're using it, they want to know what's in the water regardless, and to give them a little more perspective. So our group's also been doing this for BTK for the last 4 years as well. As we actually go, that's my intrepid technician, Keegan, on the left, sitting in one of the reservoirs in a river tied to a line so he doesn't float away. He goes in and does a lot of the water collecting, usually a day before the treatment, the day after the treatment, and a couple weeks after. And we get it tested at a lab here in town, and they tell us how much BTK there is in the water. So I'm going to try to put this in perspective for you. What we have here is colonies per milliliter. That's how they measure how much BTK there, or how many any of bacteria would be in water. These are the highest levels that have ever been tested for BTK in laboratory conditions. So these are sort of a standard battery of tests that we use. So the highest they've ever tested would be for rats, 120 million colonies per milliliter, a little bit less for freshwater fish. These aren't the lethal doses. These are just the highest levels they've ever tested, and they still found no effects on physiology, no mortality, no sort of skin conditions, no nothing. It just was really just not reactive with the species they've tested. Quebec's been doing these measurements for years. These are all the different locations on the bottom that we've done it for. I actually have it on here. It just doesn't show up based on the scale. The highest we've ever found is roughly somewhere between 400 and 800 colonies per milliliter. To put this in perspective, this is the CN Tower in Toronto. If the CN Tower represented the highest levels ever tested against rats, the levels that we're finding in water would be represented by 2 stacked pennies next to it. Or if we were to convert that into US currency, the Empire State Building and 2 American pennies. It's basically the same in terms of height. And so that's the approach we've been using in terms of, like, as a part of our communication to try to explain what we're actually using out there. And we've been pretty effective with it, I think, so far. This was from 2015. There's people that were up in Campbellton when we were first starting this work that were very concerned. You can read the quote. I'll just read the last part. They were very comfortable after seeing the work we've done, including the water testing and these explanations. They appear to be approaching this from a very responsible manner. And that was from the city, the city manager. So I think we're doing some good things. I mean, it's sort of an ongoing process. Every year, I'm out giving talks of this variation. We also have one for tebufenicide, which I just didn't really have time for today, but it's really the same sort of flavor, explaining how it works, explaining what we're finding, explaining what that means. And I think it's been very useful for people to asking those questions and that are rightly concerned about what we might be using. So that's pretty much it. I mean, I just like to, there's a lot of different aspects to developing a program like this. It requires a ton of legwork. And I think I've been working with Brian Roth and Allison Kanoti as we sort of move forward to the stuff, other people as well, trying to get the starting point for the monitoring in Maine, the outreach and talking to people. And this is a good example for that as well. I hope this talk gives you some idea of the kind of challenges or issues you might deal with as you're developing this. And anyway, so far so good for our stuff. And I guess right now, just for people that are wondering, we're coming to the end of the 4-year ACOA that funded this project. And we're waiting to see whether another 4 years of funding will come forward or whatever that might be to continue the work that we're doing. But we think the results give a good justification for continuing to develop that work. So I'd like to give a big thanks to Brian Roth, who invited me. Jenna did great work putting this together, and I really appreciate her navigating me through this Zoom thing. We did a practice run on Monday, so we made it through. Thank you to Jenna. And Allison, of course, for being both a collaborator, but also for setting up the talk for today, and to everybody else who signed up to listen to the talk as well. So I'll also thank my colleagues. My collaborators are on the bottom. Second one from the left is, for those of you who don't know her, is Emily Owens. She's our coordinator for citizen science. And you can get for Budworm Tracker, you can get that information on the bottom if you're interested in that. And of course, we have Veronique and Deepa, Patrick James, all great collaborators. And then me, and these are some of my technicians and two little boys and a few dogs running around as well. So if you guys have any questions, I'm more than happy to take them.
Great. Well, thank you so much, Rob. That was a great talk.
Thank you.
So everyone should have a group chat window on their screen. I think it's in the bottom right. You might have to do some navigating with it. But if you have any questions, we'll be here. So feel free to type them in and I'll read them out loud.
I kind of fed them off the fire hose on that, so it's fair if they need to digest it.
Okay. We're getting comments that this was very informative and that the discussion of BT was good, and they like that.
Good. Yeah, that's actually one we've given a lot of times around in our area. We pretty much, I give it 4 or 5 times per. I'm going to be giving it next week as well for some of the new councils. Oh, okay, there's a question. Mm-hmm. Is it a definite expectation that we'll have a serious outbreak in Maine is the question. It's not necessarily, and this has been sort of a subject of debate. I didn't go into some of the other climate change work that's been going on by some of my colleagues. Deepa Piraswaran in the previous slide. But the outbreak where it started this time was way farther north than it started during the previous outbreak. You know, previous one, we always had it sort of humming around in the middle of New Brunswick, and then it moved up. As of this outbreak, it started in the lower, in Baie-Comeau area, which, you know, hundreds of kilometers to the north. And so one of the questions that we debate, it's part of the reason why we collect this work a lot, or collect these data in the south a lot, is because we're trying to understand to what extent we are in a southern range limit here. And that includes Nova Scotia, southern New Brunswick, and Maine would probably be part of that as well. So it's not certain. Part of the way we collect data is trying to understand if there's things other than just what we're doing that's leading to these populations to struggle in these range areas. I do think it's safe to say that we are in something of a range area. It's just not clear how sharp that's going to be, or whether you still can have a lot of pressure with migrants pushing things to the south.
Okay, we have a few more questions.
I'll let you ask the question.
Okay, it doesn't matter, but I have one question. Is New Brunswick monitoring the effects of BT on non-target Lepidoptera, and do you avoid spraying sensitive areas?
Yes, that's from Mark. Yes, we are, part of the work that we're doing in my sites, this non-target work, everything that we collect in our sites is frozen and kept for the analysis to look at actually how the densities of Lepidoptera, not just for the parastoids, but also for densities of Lepidopterans over time, trying to understand how those dynamics are also changing with budworm, but also changing with respect to the different treatments. Some of my colleagues have also, and this will be continuing into the next work if we go forward, are also looking at Lepidopterans in terms of like light trapping. I know there's been quite a bit of work done for this with gypsy moth, because of course they're using BTK often for parts of their slow the spread program. They didn't find enormous effects on the Lepidopterans, partly just because the period during which BTK is used is relatively specific to when budworm is right at its peak of feeding, and a lot of other Lepidopterans feed on sort of either side of that. They either done earlier in the season, or they're done, they start or done later in the season. So there hasn't been really strong evidence for really significant effects, but it's also worth noting BTK isn't 100% efficacious anyways. It's still relatively mild. It takes sort of a percentage of the population, but not in the way that a lot of the past chemical insecticides did. Let's see here. Oh, and do we avoid spraying sensitive areas as well? We tend to be, right now, I think we've been working in most of the areas where budworm has been present. There has been lots of discussion, especially in areas like PNAs. What do we do when we go to parks, like, for example, Mount Carlton and this sort of thing? And this is ongoing discussion, I think, among sort of the provincial people with others about what do we do if we're trying to control hotspots. And part of it is just a discussion with those groups to see what their priorities are for those areas. So I guess that's an ongoing discussion. Oh, let's see. So here's a question from, or a comment from Ram and a question. Most people won't pay attention to the outreach and explanation until the outbreak reached epidemic levels, and then emotions prevail. And any ideas how to counter that? That's a great point. And that's actually was a center point of our discussion when all this started 4 or 5 years ago. You have to remember, 4 years ago, we had probably just those little tiny, maybe patches of defoliation like Maine's describing right now. So if that also gives you an idea how fast it could move forward in Maine, it may give you some ideas. I mean, that was the biggest issue. I think we've just been very proactive, and I mean we, the province, the stakeholders like the forestry companies and everyone, because there was an option potentially to do some containment. You can only do that when populations are still low and before it starts. I mean, Quebec can't do anything like this because it's way too late. So I think there was a really strong push from a lot of people that supported developing the idea to move this forward. And that really is what it is, this proactive approach with communication. Not everybody is going to listen, but I think, I mean, you can only reach out to the people that want to understand. And when it comes to budworm, people do, especially forestry companies or people that own woodlots or people that visit parks, do want to know what can be done, at least what the options are. So I think just sort of continuing that communication is really the only way that you can do that. Tom Charles, many years back, Dr. Diamond commented that spruce budworm was nature's response to an outbreak of balsam fir. That actually was one of I didn't talk about it. I should have squeezed it in because it looked like I had time. There's the first ever hypothesis that was introduced to explain spruce budworm was this silvicultural hypothesis. Okay, and this was the idea that they're in the early part of the century, they were cutting huge tracts of spruce, and these were replaced by balsam fir. And that's true. That definitely does happen when you have balsam fir that comes back. It's usually what comes back very quickly in a lot of the places that are cut. But there was all sorts of things that sort of had a lot of traction for about 50 years before they started revisiting sort of dynamics involved with predators and other types of things. It just didn't really match with a lot of the pieces of evidence. One of the parts was, so if you had large tracts of, or if you basically budworm came around when you had large tracts of over-mature balsam fir that sort of led from a harvest cycle. It took about 70 years for budworm or for balsam fir to reach that point, and budworm outbreaks were occurring on 35 to 40-year intervals. So the budworm outbreak interval didn't really match the forest sort of cycle intervals. So that part didn't really match. The other part was that we had pretty reasonable evidence of budworm outbreaks long preceding when like forestry practices started removing balsam fir. So that sort of, and I should point out that that doesn't mean that forest composition isn't important for budworm outbreaks. It doesn't drive cycles maybe, but it may influence intensity. So if you have forests with large tracts or with large densities of black spruce, for example, it's a less favorable host, or with higher infusion of like deciduous hosts or more of a mixed type of forest, there tends to be a more complex, well, more resistant in terms of foliage quality if it's black spruce, but a more complex sort of dynamic natural enemy web in more mixed forests that can keep budworm populations lower. But one of the things we've also noted in the last 10 or 15 years of research on that question is that when budworm populations get really high in an area, those things don't matter. All the trees end up getting defoliated anyways. So at lower levels, you see really strong effects of forest composition, but at high levels, it tends to overwhelm other sites. Let's see here. Ian Pryor, do you think mitigating activities being deployed in New Brunswick will in fact reduce the spread, timing, and intensity of spruce budworm in Maine? Well, I think one of the encouraging things so far, at least as far as Maine is concerned, that most of the issues that we've been dealing with in New Brunswick have been fairly concentrated over to the east. You guys probably saw on the map, there's the little patch we've been treating up in the northwest corner there. It doesn't seem to, it's there, it's getting defoliated, it's getting treated. It doesn't really, we're not really seeing the same sort of pattern or extent of spread in that corner. I don't know if that's because the way the weather patterns go, it just keeps pushing everything towards the east. But we are seeing sort of this asymmetrical development of the outbreak in the province so far. I think there's sort of two ways that the work could help Maine. The first would be that it at least ends up being a proof of concept. You're going to be seeing these treatments, I think, in New Brunswick, providing funding comes through for the next, 4 years at least, or as long as the outbreak takes place. Ideally, keeping things at bay. That, at least on one side of Maine, helps to keep things from moving over to Maine, especially if it's in northern New Brunswick. It may give a proof of concept that gives us at least some ideas that Maine can adapt for how they might want to manage it, if they do decide that they want to. I think, the conversation I've had with people in Maine is mainly, if you get the communication started and you get the monitoring started, you can have a discussion of what you do when it actually becomes an issue later. And I think as long as they're on track with those two key things, which they are, I think the other decisions can be made when they need to be made. Let's see, Gary Fish. How often do you get questions about BTK and the questioner wonders if it's the same thing in GE, corn, or soy? It is. BTK, at least the toxin in BTK is what they integrate into GE, corn, and soy. But I've never had that question, so this is the first. So I guess you're going to see how I answer that off the cuff. I would say, this is not a genetically modified species, or at least what we're using. Yeah, I guess I would just probably the way I would answer that would be just to distinguish those two, and so not to conflate this with conversations around genetically modified organisms and this sort of work. I think sometimes it's better just to not, you can help just by not conflating those two. Yeah, no kidding. I'm surprised it has never come up. Yeah, that's a great point. Maybe somebody will ask me. I should probably read a little more on that too. I do know some things about it, but because I usually only respond after I've been smacked with a question. So maybe this is my wake-up call.
All right, well, with that, it's about 3 o'clock. So I think we'll end the webinar. Thank you to Rob and Allison for presenting, and thank you to all of you for your interest and for attending. Please check your email in the next day or two for a form that I'll fill out. You need to fill out this form to get credit for the webinar. And we hope that you join us next time. Thanks again.
Thanks, everyone.